let's continue on. So, having seen that there are 20 basically amino acids, different amino acids are there, the way they are assembled right now, I mean, this is the kind of a snapshot cartoon of explaining basically roughly what's going on during the protein synthesis. Right now, by just one way of practicing ourselves in many different figures in biology is just by looking at, trying to, assuming that you've never been exposed to this type of figure, but still, how much can you, just by looking at for the first time, how much can you pick up out of this figure? Because the one general rule of any biological figure is grab a whole picture and then try to individual detailed label. For example, oh, what is this? I mean, sorry. For example, what is this? And what is this? And then you try to assemble your thoughts, figure out, okay, so what this? Okay, this is the whole thing is about the ribosome. So this whole big thing, bulky thing is the ribosome that I mentioned, that the whole structure within which the actual protein synthesis is going on. Once again, I emphasize, you do not have to know any details about this, and maybe even not a single thing about it, but just try to test yourself. Okay, by looking at this, how much can I just figure this thing out? Maybe you may even be surprised of yourself that, oh, surprisingly, you can just pick up so many different information simply by trying to figure out, you just pretend that you have to. You're not you are trying to teach this thing without without being prepared. Then just simply trying to looking at this figure and try to make up any explanation or story. And then a lot of times actually you will be really surprised at how much can you pick up and then be able to deliver. And then a lot of those things are actually turned out to be correct information. That's how you can approach. One beauty about this biology is a lot of things can be actually communicated through this type of graphics. Yeah. That's one thing personally I like about it. But anyway, this whole thing is ribosome, and each ribosome, what you can see is what's going on over here, and you see the labels. Messenger RNA. mRNA is one type of RNA. And this is the kind of exact copy of the DNA part, the gene part of the DNA. And if I forgot mentioning the difference, the fundamental difference between DNA and RNA is, whereas DNA usually exists in double-stranded through this complementary base pairing, for some chemical reason, RNA loves to be alone. I don't want to. That doesn't mean that RNA cannot participate in this complementary base pairing, but for some reason, chemical reason, RNA rather stay alone as a single strand. So that's why you see only one strand. RNA usually exists in single stranded. That's why in the earlier slide, the figure comparing DNA and RNA. DNA portrayed itself as a chubby, big fat, because it's double-stranded. And RNA is a skinny one, because usually it exists, not always, but usually it exists as a one single strand. And here, this RNA, the copy, exact copy. So that means this RNA who gave this fundamental information about the protein the amino acid sequence in a given protein, simply copy one of the two strands of the DNA, and that serves as the information for particular protein. But anyway, so this whole RNA is laid upon, and basically what this whole ribosome does is read through, while moving along on top of this RNA, messenger RNA, mRNA, and then moving along each different part. And while doing so, here in small area, small chamber within the ribosome, other type of RNA is called the tRNA. This tRNA they are different from this mRNA in that the function of this tRNA is to bring carrier. They are acting as a carrier of individual amino acids. In other words, you can easily figure that there's got to be at least, in the cell, there has got to be at least how many different tRNA exist. If we utilized 20 different amino acids in this protein assembly, at least how many different types of tRNA should we have if we say each different tRNA acts as a carrier of amino acid? Each different type of tRNA. So at least we should have 20 different transfer RNA, but we have actually many more than 20 different tRNA for some reason. But right now we don't have to worry about that. So each different tRNA can bring different amino acid onto this site and roughly say what's going on. What do you think is going on over here? While two individual tRNA carrying this amino acid are sitting over here, one amino acid is being handed over to the other guy. And the reason why this tRNA has so many amino acids is the end result. Because each of this tRNA coming in gave its own tRNA. Hey, here, have this. And that's how one particular tRNA ends up whole chain of amino acids. And then eventually it will stop somewhere. And then that we call a protein product. And that's how we do this basically protein synthesis on this ribosome structure. Okay. I only very sketchy, I gave you very sketchy description. Okay? Because actual real detail you don't really need to go into. But simply, whenever you get to see this type of picture, photograph, or whatever, then from now on, they are just the same old story of ribosome acting on the protein synthesis. That is just good enough. Here is another such description. Now, the only important thing about this protein right now, at this moment, is this. So with this 20 different amino acids as a random assembly, the beauty is you can generate so many diverse shapes. I always emphasize these shapes, okay? Because shape exactly related with the function in biology. Shape will dictate the function, and protein is the only macromolecule out of 4 different macromolecules that we have studied. Protein is the only one, perhaps the only one. Maybe RNA can try to mimic. RNA is the other, second type of such thing that can generate us a little bit of such different shapes. But forget about the RNA. But protein is by far the only type of such a big macromolecule that can generates so many different types of shapes. And that is the reason why now protein is the one who can take care of so many different diverse functions, like enzymes. Okay, you know what the enzymes are? Receptors. Okay? Receptors are something that it has to recognize some small molecule and then bind, and that kind of signal must be delivered. It's a receptor. And even transporters, antibodies, right? Remember those antibodies? And other structural proteins, etc. Like all different protein structures are, in these days, scientists have figured real exact structures and then showcase their shapes in 3D modeling like this. So antibodies shape this. Usually we depict this antibody as a Y-shaped, and enzyme has so many different enzymes has so much different types of shapes and hormones, etc. A lot of those different things. Okay. And some of those are structural proteins, as you can see. Your fingernails, feathers, and spider webs. They are all different types of proteins, and hairs. So think about all different, how many different ways can you use protein? And it's all about their different ability. Their ability to generate different structure and different shapes is the one. And
enzymes are proteins that carry out chemical reactions. They have two special features. First, they are highly specific. Each enzyme acts only on select molecules called its substrates. Second, enzymes are catalysts. They greatly accelerate the rate of reactions they carry out, and they are unchanged by the reactions they carry. Enzymes don't get used up by chemical reactions. They carry out the same reaction over and over as long as their substrate is available.
So here
enzymes have a recessed pocket on their surface called the active site. The shape of this pocket precisely matches the shape of the substrate. Molecules that don't bind snugly in the active site don't react. The shape of the binding pocket is the key to enzyme specificity. This enzyme is chymotrypsin, which breaks down protein molecules in the stomach during digestion. Note how precisely the large side chain of a specific amino acid fits in the depression within the binding site. That precise fit positions specific atoms of the substrate for reaction with atoms from the enzyme.
The point
Enzymes work by lowering the activation energy of a reaction. Biological molecules are generally very stable. They don't spontaneously break down because the activation energy for that reaction is too high. Enzymes lower the activation energy by positioning the substrate in just the right shape at just the right distance from a reaction partner for the reaction to occur in the active site.
So are you over now? Finished?
When the reaction is completed, the products diffuse out of the enzyme. You might wonder why the products don't stick in the active site, since the substrate fits well. It is because the products are now different molecules with bonds and electrons in different locations. The active site binds the substrate with just enough energy to promote the reaction, but the product diffuses freely away. When the reaction is completed, the enzyme is exactly as it was before the reaction, ready to react again.
Okay, are you finished? Okay, good. Now, how much did you pick up right now, this whole short movie? Can we share among us, like, what kind of keywords that's stuck on your head out of this short movie? Like, let's name some of the imprinting keywords, vocabulary. In that way, we can somehow figure out how much could you understand about this. Any particular vocabulary that you have lingering in your mind out of the short movie? What? Active site. Thank you. Yeah, active site. That's a good thing. Active site. That means enzyme. What is an active site? It's the site where the enzyme actually recognizes the substrate. So always there is a relationship. As for any kind of enzyme, any enzyme will have a particular relationship with what? It's called a substrate, yes. So this relationship between enzyme and substrate is always had to be this kind of very special fit relationship in terms of a shape. Okay? So enzyme substrate. And similarly, in the case of receptors, receptor is not an enzyme, right? But receptor has also very specific relationship with something called ligand. This is another vocabulary. Ligand is a very specific small molecule that specially fit into any specific type of receptor. Okay. And in the case of antibody, what about it? Antibody always has, special relationship with it, antigen. So if you think about it, the common denominator between all these three different types of association or interaction is always a shaped driven. Whether these two parts have this kind of fitting shape or not will dictate this relationship. Whether it's an enzyme-substrate reaction or receptor-ligand reaction or antibody-antigen reaction. And the results are only different. They are all will have to start this fitting thing. But in particular, in the case of an enzyme substrate reaction, the only difference is now that interaction occurs in the active site, right? The special. The enzyme, maybe the whole entire enzyme molecule will be this big, but there might be only a very small area where this area will provide some fitting interaction with particular substrate. That's what we call active site, right? But the next thing is this enzyme substrate reaction, the uniqueness about this thing compared with the antibody antigen and the receptor ligand is this thing is what? This is some chemical reaction is going on. Unlike the antibody antigen, they just simply bind. That's it. Okay. They just interact and then grab and that's it. Receptor ligand is usually also the binding itself is the end result. But in enzyme and substrate reaction, they bind and that's only the beginning. What's next? As a result of this binding, now actual change in the substrate. That's what we call the reaction, chemical reaction. The substrate the shape of a substrate will change into something else. So then now what? That's why you saw some release of the substrate out of the active site. So here the important thing is substrate should not stay in the active site for a long period. They should stay there very immediately and then leave. Why? As a result of the chemical reaction. What happened? As a result of chemical reaction, what happened? The substrate shape changed. So once they fit into this active site, so that's the reason why this particular substrate was able to fit into this. But then as a result of chemical reaction, now the substrate molecule has changed into something else of which shape doesn't fit into the active site anymore. So they have to leave this room because they don't fit into. And that's the end. So they release. So they have to keep cycling this. The sooner each enzyme can do this rotation thing, and then we regard usually that enzyme as a very good enzyme because it completes this chemical reaction fast. If this whole thing, changing takes, for some reason, relatively very long, then we consider that enzyme is very poor enzyme. Hey, you are not doing very good job. So whenever I find something better, then I will just replace. I will kick you out. I will fire you, kind of enzyme. So this speed is always dependent upon how quickly you can make a substrate fit into and then change that substrate shape into something else and then release it, is the ability of any particular given enzyme. And what else? What else did you pick up out of this short movie? Do you still remember something like this? Activation energy? Yeah. What is this? What is this activation energy? That's something that the major point that I'm going to go over in the second part of this. Because this is all about now. Okay, so once at least you remember activation energy vocabulary, so good. Now we can talk about this thing. So once again, let's try to put everybody on the same page. Enzyme is biological catalyst. What do you mean catalyst? The catalyst means it actually is not being consumed in the actual chemical reaction, but it still stays there as a kind of neutral guy, but the only thing it does is speed up the rate of reaction. So the enzyme is what? That's their job. That means one single enzyme can be used many times, to repeat a chemical reaction, because the enzyme itself is not being used up. It simply speeds up the chemical reaction. Another thing is, so it is not consumed in the reaction, it only speeds up the reaction, is what it means, the catalyst. So in chemistry, to make any particular given chemical reaction go faster, we usually frequently use such a catalyst. Okay? But in biology, in a living system, these protein-based, whatever those substances, serve as this kind of a catalyst. So that's why we call enzymes as biological catalyst, right? And although not all the enzymes are made of proteins, but usually the vast majority of enzymes are solely made by protein, and you know the reason why. Because proteins are the one only can provide such complex different shape, each of which should be able to fit into particular different substrate shape. DNA cannot do that. DNA cannot even think about it. DNA is such a boring material. Carbohydrates, it's difficult for them to, for some reason. Lipids, no way. Maybe RNA, some RNA actually. To a lot of surprise, the scientists actually, initially it was such a big surprise in mid-1980, 1985 actually, for the first time, two different scientists in the United States independently discovered actually that not only proteins but some RNA can act as enzyme. It was such a big shock. And Obviously, those two scientists won the Nobel Prize because of their work. Because the fact that RNA can also act as an enzyme was such a big impact. But still, majority of all these things, enzymes, the functions are provided by protein, and because of the reason of these things. But now, this question: spontaneous reaction. Spontaneous reaction, by definition, is something that you don't have to worry about it, because it will occur. It will occur in itself, and then it will complete without doing anything from outside. That's the nature of a spontaneous reaction. For example, if we eat our meal, basically mainly made of glucose, okay, those glucose molecules will be digested into smaller fragments, and by doing so, the energy will be released so that we can use that energy in building up the ATP. So at least this breaking up this glucose into smaller parts is a spontaneous reaction by nature, right? So it will occur once we eat. Then each glucose molecule, and then go through the digestion, so that as long as we present those our meal in the individual glucose molecule, then that glucose molecule will be degraded into smaller one. But still, in that case, do we still need an enzyme for such a chemical reaction of degrading the glucose into small inorganic compound, like eventually like carbon dioxide? So that's what we do. We eat and then break them, and then as a result we exhale the carbon dioxide out of it. Okay? And we need the oxygen for that particular process. So that's why we cannot live without the oxygen for that energy release, energy production purpose. But anyway, so just think about this. Why do we still need an enzyme for such a spontaneous reaction? Because without the enzyme, without the intervention of enzyme, still the chemical reaction will occur. So why do we do it? Why bother? The answer should lie in what? The fact that enzyme is a biological catalyst, isn't it? That means it will speed up. So how much faster can it make, practically speaking? So let's say we got sick and tired of being bothered by enzyme. So we decide to, from now on, I will just eat my lunch, and then hands off the enzyme that will just take care of this whole breakdown. Hands off. It will break down itself. I will wait. Okay? I will wait all this natural process. All those enzymes go away. And how long will it take? If we just eat our meal, the glucose will degrade so that we can actually extract some energy out of this glucose molecule without the involvement or without the help of enzyme, because it will occur anyway. So how long will it take? Let's say with the enzyme, probably that will be completed in like something like 1/100th of a second. It will take much less than 1 second actually to finish that. But without enzyme, how long will it take then? Maybe 1 minute? 10 minutes? What do you think? 5,000 years. So that's the reason why we need an enzyme. No one can wait 5,000 years after you have lunch. So you better, although I don't want to, but we better let those enzymes take care of this whole thing. Even though it is a spontaneous reaction. That's what we are talking about. And that is something to do with so-called the activation energy. Why without enzyme it will take so long? Because of this activation energy. So even a spontaneous reaction, this catabolic reaction means those breaking down reaction, which is a spontaneous reaction. So glucose degradation is one very good example of a catabolic reaction. There are only two different kinds of reaction. One is a catabolic reaction. Starting from big molecule, you end up with smaller molecule is called catabolic reaction. The opposite word of this catabolic reaction is an anabolic reaction. Anabolic. Okay? Anabolic reaction always occur from smaller, starting from a smaller molecule component, and then you try to build larger, more complex molecules. So anabolic reaction will be always non-spontaneous, energy requiring, whereas all those catabolic reaction will be spontaneous reaction, and it will release the energy, right? So anyway, so this catabolic reaction will be synonymous to a spontaneous reaction. But still, in our cell, those catabolic reactions like a glucose breakdown cannot occur by itself, practically speaking, because it will take 5,000 years. Then who can wait 5,000 years? Due to this so-called activation energy. What is it? This activation energy is diagrammed like here. This like camel's back like a hump. So the reason why this whole thing can be regarded as spontaneous reaction is the starting point, the reactant. The starting material has a higher energy than the product. So in other words, at the end of the reaction, this is the progression, the reaction progress. At the end of the reaction, this whole product will have a lower energy, which means where did all those energy go then? It was released. So that's another way of presenting the characteristic of spontaneous reaction, right? You have a big molecule, starting point. You have a big molecule, like glucose. And you digested the glucose into individual, very small part. And what did I say about it, the reaction? Then energy will be released, right? That's what exactly, in chemistry, they love to describe such thing in this diagram, in chemistry, in biochemistry. Right? So, starting molecule has a higher energy. This, and then end product became much lower energy status. Means all these energy differences, all this energy were out. They were released. Isn't it the characteristic of spontaneous reaction? Yes, it is. So, okay. So this is, whenever you see any energy diagram like from uphill going downhill, then you know, that is something about a spontaneous reaction, which is, another way, catabolic reaction, isn't it? Catabolic reaction or energy releasing reaction will always be described like this, from going up to downhill, right? And it's like, practically in practice, it's kind of a more, the analogous to on the hill, on top of the hill, you try to roll any rock, and it will go spontaneously. So easy to roll down to the bottom. Is very similar to this kind of. However, what you see is this. What do you see over here? This hump. What is this? So without this hump, any product will just go automatically, will be rolled down into. But practically, each different chemical reactant possess this energy barrier. to start off. To start off any chemical reaction, you need some kind of a seed money. Okay? But it is not really easy to bring up those seed money to overcome this huge energy barrier. And this is called activation energy, the kind of energy that required for starting any chemical reaction. It's kind of, what is such a vague concept, such an abstract definition? What is it really? In reality, what is the activation energy is, chemical reaction is what? In order to get any chemical reaction going, chemical reaction is nothing but changing, rearranging the chemical bond. So even though the final product, the chemical bond rearrangement is much more favorable energetically, so that at the end you will get the huge reward of release of energy. But in order to get this thing started, you have to break. You have to break and rearrange the existing molecule, and they will severely resist. Hey, what's going on? I'm going to stick with this. And then to take this thing apart and then try to rearrange is not going to be easy. Okay, so that's kind of activation energy. It will require some input, initial investment of this whole energy. And usually, without the involvement of the catalyst like enzyme, it is very too much of a big barrier. But enzymes, if there is an enzyme, then it will. Enzyme can reduce this whole thing into manageable amount. That's what this enzyme can lower the activation energy. And practically, what this whole reducing the activation energy can give us is speeding up. Like something that theoretically you calculate it will take 5,000 years, but it will be done in the presence of an enzyme, because it can simply press it down so that it can occur even less than a second is what this enzyme does. So, saying all these different jargon-like things, the only take-home message of our thing is, the reason why enzyme is needed for any spontaneous reaction is it can reduce the activation energy so that it can make the chemical reaction much, much faster. That's all. At this point, if the enzyme and substrate interaction was based upon covalent bond, what do you think it will happen? All these enzyme substrate interactions are what? All those non-covalent bond like hydrogen bond, ionic bond, or van der Waals interactions that we have learned in previous lectures. But instead, let's imagine if one brave enzyme came up and declared that, hey, I'm sick and tired of having to deal with van der Waals force, what is this? And an ionic bond, I don't like it. So I want to use more stable, strong covalent bond for me. Good for you. Okay, so what do you think? That enzyme will become very stable? Maybe very stable, but it will become obsolete because it will take forever for that enzyme to finish off one task. Because chemical bond, the covalent bond is very, very difficult to break and also very difficult to establish, unlike such non-covalent, very weak bond. That is the reason why biology so much relied on those very weak bond like ionic hydrogen and a van der Waals bond like this. Because it's very weak, So it's very easy to break and very easy to reestablish. So you can keep this thing rotated so quickly. But covalent bond cannot keep up with this. So anyway, so that's one of the reasons. Then what about this thing? So the enzyme, the business dealing with the enzyme, we have two different sides of story that we need to take care of and understand. Okay, in the case of this spontaneous reaction, okay, I can understand the reason why enzymes are still needed, because it can speed up the chemical reaction. But totally different issue over here. Now, non-spontaneous reaction, like this anabolic reaction. You have a totally different problem you have to solve. This is not going to happen. It's not supposed to happen because it is a non-spontaneous reaction. How are you going to do this? What did I say about non-spontaneous reaction? It can happen as long as you provide the energy. It's a very good way of saying it, but how? How are you going to provide the energy? You're not there and watching over this reaction occurring and then pour some energy here. This is an energy. You're not doing it. Somebody else. So enzyme. Do you mean enzyme can do this? Enzyme somehow bring the energy, and here, the energy? Maybe, somehow. But this whole energy requirement is totally different thing. Just because you are an enzyme, like you may actually imagine enzyme is just kind of like a magician. So even if this is, for example, even if this is this kind of reaction, which is, now if this is a starting point and this is a product and this is a reactant, and is this a spontaneous reaction or a non-spontaneous reaction? Non-spontaneous. It's uphill, right? So this is non-spontaneous reaction. So it's not going to happen. But hey, if we somehow invite enzyme, then enzymes are such a nice guy and a wonderful guy. So maybe enzyme can do this, restructure this thing into this. So enzyme can do this? No. This is a universal rule. Enzyme cannot change any particular dynamic of any given chemical reaction into favorable one. You cannot touch. Then how? Then how enzyme can make this non-spontaneous reaction possible? Is the question actually. A very fundamental, important question. And every studying biological and chemistry student must learn about this. The examples of anabolic reaction have this kind of a list. It is a synthetic reaction, characteristically so, from simple component to build a complex reaction like this. And by doing so, it will result in the decrease in the entropy, so it is non-spontaneous, right? So energy input must be done. The question is, who and how can provide this energy? Then enzyme can do, of course. Enzyme will be able to somehow provide the energy, but how? It's called energy coupling. Enzyme acts as a kind of a matchmaker, a pickup. So enzyme sees, oh, this is a non-spontaneous reaction. Oh, poor guy. So this reaction cannot occur in itself. So how am I be able to make this reaction happen? somehow I can provide the energy. But how? In what exactly? Energy, providing energy itself is a very vague concept. Exactly how practically you provide energy is something that needs some more concrete explanation. What the enzyme does is bring a naturally spontaneous reaction together and then make them happen at the same time. That's what enzyme can do. Because enzyme has this active site, isn't it? In which chemical reaction can occur. So all it can do is bring one such substrate which requires this non-spontaneous reaction, and then on this other side, bring up one chemical reactant which can go through this natural spontaneous reaction, and then make these two chemical reactions occur together at the same time. And so this is one uphill reaction, and this is one downhill reaction. If these two chemical reactions occur together, then you have some differentials, energy differentials. Although you may require a certain amount of this energy requirement. However, for example, ATP breakdown is such a very good example of such a downhill spontaneous reaction. You can generate this much energy by using this ATP. And that is the reason why ATP is regarded as a what? Energy currency. You bring up this ATP. So enzyme can bring the ATP over here and break it down. Okay. And then you get this huge energy released. And you do this ATP breakdown together at the same time with any kind of this non-spontaneous reaction. So, hey, I need this energy. In order to drive this chemical reaction, I need this much energy. No problem, Jose, because I have this much energy will be released right now. So you do the math. You subtract this from that, and then you have this gain overall, whole net energy gain. So this whole overall reaction will occur spontaneously. And this kind of a coupling thing we call energy coupling. So once again, bottom line is enzyme can do this energy coupling. In other words, enzyme can make any non-spontaneous reaction possible by energy coupling, which means coupling one non-spontaneous reaction with one very big spontaneous reaction, so that overall energy will become spontaneous. Because the energy released from one particular spontaneous reaction is larger enough than the non-spontaneous reaction which require the energy input, just like in this case. And usually the ATP is utilized in such occasion as an energy provider. That's why ATP is utilized as a universal energy supplier in the cell. So that whenever they are needed, enzyme can pick up ATP and then do this, convert this ATP into ADP, then you get this huge energy released. And some of them can cover up this energy requirement. And then still you have left over, which make all entire combined reaction overall a spontaneous energy releasing reaction. Okay.